I am now excited to introduce you to our next speakers who are investigating new research models of low-grade serous ovarian cancer by using patient tissue samples. Joining us this evening are Dr. Carey, a gynecologic oncologist at Vancouver General Hospital. His primary areas of clinical and academic interest are the management and study of rare gynecological cancers, preclinical drug evaluation, and predictive biomarker discovery and application. And working with Dr. Carey and joining us for the presentation this evening are Dr. Marta Llauradó Fernández, Dr. Nelson Wong, and Hannah Kim. So within Dr. Carey's lab, Dr. Llauradó Fernández is the research associate and program manager, and Dr. Nelson Wong is the staff scientist. So I will turn it over to you, Dr. Carey.
Okay, can everybody hear me?
Yes, we can hear you.
Okay, great. Thanks very much for having us. We really appreciate the opportunity to be part of the meeting today. And yeah, we've really heard from some excellent researchers already. So and it's been nice to see how they've really highlighted the need to do patient-oriented research. We're really fortunate to be part of this journey for patients and individuals affected by low-grade serous cancers. And so we'd like to share some of that with you today, some of the work that we've been doing. And we really appreciate also the support we've received from Ovarian Cancer Canada. So it's great to be here. There's a couple of other members of the lab, Maddie, who's here, who's been our master student, and Yun Yi, who's now one of our technicians, have joined us as well. So nice to be here. Yeah, there they are there. So we're going to talk a little bit about the work we're doing. Can I control the slides here? Is that, yeah, great. Yeah, so this is our lab, and it's been a great experience. We've been working at this over the last 10 years, I would say, trying to advance and improve treatment for patients and individuals affected by low-grade serous cancers. So we'll talk a little bit about that today. And this is a group we've already, you'll meet some of them directly. So to introduce you. So for ovarian cancers, one of the transformations that we've made, and this is how we identified in part that low-grade serous cancers are a distinct type of cancer. So we recognize in ovarian cancer, there's many different types of cancer. We used to characterize them just based upon what the cancers look like by light microscopy. I don't know if this works here. The pointer, yeah. So these are the light microscopy. So a pathologist would look at the slides and just tell by light microscopy sections under the microscope what type of cancer it is. But now we appreciate that even within each type of these cancers, there's distinguishing molecular features that characterize them, and some of them are more similar as different types of traditional cancers than they are different. So we now not only appreciate what these cancers look like under the microscope, but these unique molecular features that characterize the cancers, and now we can use these to determine different treatment strategies. So in low-grade serous cancers, this is the group, they tend to occur in younger women. And then there's a spectrum of different mutations that we see in these cancers that distinguish them from some of the others. So we've been really fortunate that we've been assisted, our work's been assisted by many patient advocates and families of individuals affected with low-grade serous cancers. And they really helped get our program started, and Ovarian Cancer Canada and all the work that they are now doing on research for ovarian cancers really helped us do a lot of this work. And so I just want to recognize some of the patient advocates that have helped us, and I'll talk a little bit about them later, more specifically. And when it comes to, we've already heard a little bit about, oh, okay. Sorry about that. We've already heard a little bit about the need to understand or ask the best questions as we understand the patient experience. And so in low-grade serous cancer, patients have really told us about how important it is to have equity in their cancer care. They have fewer treatment options than the more common types of ovarian cancers. There's less research that's been done. There's less research funding, and so we've worked hard to try and correct some of those inequities. And patients have clearly communicated to us how important it is for them to have more treatment options and more effective treatments for their cancer. So these are the learning objectives of things that we were asked to address today, but we want to talk to you a little bit about some of the work that we've done on the rare gynecologic cancer database and low-grade cancer specifically. We got this started. Where Marta is going to talk about some of the laboratory models and the types of research models that we've been working on that help facilitate research. And then we'll give you a few examples of some of the patient-partnered research that we've been working on. Hannah's going to talk a little bit about the database, as she's really done a lot of work on this, and we're pleased to see that we're able to operationalize some of this work now for some of the other research projects that we're working on. So Hannah?
Hi, my name is Hannah. I've been involved in creating multiple REDCap databases for rare gynecologic cancers. So we've had success with our retrospective low-grade database, which sparked interest in 6 Canadian provinces, resulting in the creation of a multi-center data sharing agreement, which includes 5 major cancer referral centers. So most of the database work was unfunded, but with the help of Ovarian Cancer Canada, we have now submitted to the Strategic Science Fund to support this. So the main goals of the rare database is to report on patient outcomes, link patients to clinical trials and research initiatives, as well as annotate models for Carey Research Institute. So the recent white paper submitted by the Ovarian Cancer Canada, we were successful in securing $1.5 million to sequence rare gynecological cancers across 4 provinces. So by sequencing these rare cancers, we can understand the biology of these cancers. So with our database, we can then link these molecularly characterized cancers to the clinical features, such as treatment information and outcomes. We can better understand the disease and look for a better treatment for the disease. And Marta will now show you what we've been doing.
Yeah, Marta's going to talk a little bit about the model development now. Thanks, Hannah.
So hi, everyone, and I'm very pleased to be joining this session today, and very impressed with every other speaker and participant and all the patient experiences. So yeah, I'm just going to focus on explaining a little bit more the patient-derived research models, specifically the ones that we have developed for low-grade ovarian cancers. And this is mainly funded by the Ovarian Cancer Canada One initiative. Now, our laboratory has been funded through the different research priority competitions, and we're going to be focusing on the One and Two for this time. So all these patient-derived research models can be used in different ways. There are 3 main ways. One is the cancer biology, studying better the tumors and how they evolve, appear, and progress. Also to use them as tools for drug testing and prediction of drug efficacy, as well as to accelerate research by sharing them with other laboratories across the world. So our lab mainly has been using tumor samples or ascites from low-grade serous ovarian cancer patients that consented to research. And we have been even growing them in laboratory Petri dishes, so some type of plates where we see the tumor cells and we expand them over time. And we also try to implant tissues on mice and try to develop models that we can keep using in the lab for molecular studies, as well as drug testing and drug efficacy prediction. So to date, we have been developed the largest collection of low-grade serous ovarian cancer patient-derived models. This includes like more than 20 different cell lines, cell cultures that they look like this. These are the the tumor cells growing in Petri dishes, as you can see here. And we have also developed 9 different patient-derived mice models where we keep propagating the tumor from the patient and trying different drug treatments. Some of the uses of these models, as we mentioned, is for drug testing and drug prediction. So in collaboration with a group in the Netherlands, we have been using some of the patient-derived cell line models and mice models to test a novel drug combination that seems to be working in a subset of low-rate patients or low-rate models. And we have been trying to identify what it could be associated with drug efficacy by looking into biomarkers that distinguish cells that are sensitive and resistant to some of these treatments. And this was published last year. Another use of the patient-derived models can be not only to test existing drugs, but to discover new treatments. And this work, one of the works we have done was in low-grade serous, was in partnership with Josie Smith, a patient partner in research, so a patient as well as a physician, and Dr. Martin Cobble, a pathologist in Calgary, with some people from our team. We basically use the low-rate serous ovarian cancer patient-derived research, cell line models to investigate what was in the surface of these cells to identify other ways to target them and deliver existing drugs or new drugs. So by doing that, we identify folate receptor 1 or folate receptor alpha that seems to be highly expressed in some of the low-rate tissues, not in some others, and definitely not in some of the normal tissues. So it allows us to target a subgroup of patients with a more accurate drug delivery method, that it's using antibodies linked to a drug that can target these surface markers and deliver the drug to the right cells and not the normal cells. So it's a more direct anti-cancer treatment approach. Now, this is a drug that's called mirvetuximab. Again, it's an antibody linked to a drug. And it has been already tested in some high-grade serous ovarian cancer patients with some promising results. Again, these treatments seem to be working in a subset of patients. But we have, through this first clinical trial, researchers or physicians identified the patients that seem to be more able to benefit from these treatments are actually the ones that have the highest levels of expression of the receptor. This seems to be the case for about 40% of the low-grade serous ovarian cancer tumors we have analyzed so far. So now there is a new coming clinical trial that it will include low-grade patients with this new drug. So we're hoping that there's going to be some good results from this study. Now, through the Often One initiative, we also got funded to develop a different type of model that it's in between what's a cell line that's just epithelial tumor cells growing in a plate, and a mice model that's a piece of the tumor growing and propagating into mice models. The in-between model is called a 3D cell culture or organoid, and basically it allows to grow cells in a more tumor structure. So it is more comprehensive of what a tumor, it has the complexity of a tumor, but it allows to test drugs much faster than in mice models. So we're hoping that we can develop with these models to faster screen some of the drugs before we go to mice or even patients. So again, another of the uses of the patient-derived research models is accelerating progress and creating larger impact. And we have been able to do that through sharing our models with the international community. Like our research was mainly funded started to be funded by a single family, the Mackenzie family, and now we have been able to, with the help of Ovarian Cancer Canada and other funding sources, we have been able to share it and expand the research that can be done in low-grade serous. One of the examples is the low-grade serous community of practice across Canada that has been established, and we are able to track clinical data across the country. The Low-Grade Serous Ovarian Cancer International Consortium is planning to expand the database into the US as well, and to do other like molecular analysis in tumor samples as well as cell lines. Then we have a couple of collaborations in UK and Australia. They are going to be doing high throughput drug screens in our cell line models, and they are going to be testing over 3,000 different compounds, single and combinations, to identify novel promising treatments for validation and biomarker discovery. And now I'll pass the word to Nelson.
Thank you, Marta, and thank you for this for the opportunity that we can share what we've been doing. So my name is Nelson, and I'm a member of the lab of Dr. Marta and Carey. And in the past, most of my work was related to developing patient-derived xenograft models. But today I would like to share with you a very special test that we were able to develop in the lab. And then this work is actually funded jointly by Ovarian Cancer Canada as well as Cancer Research Society. Now, so as mentioned, one of the major interests of the lab is to study and understand low-grade serous ovarian carcinoma, which is a rare ovarian cancer. And we know that with this type of ovarian cancer, a lot of the tumors actually express a hormone receptor that is called estrogen receptor. And we have learned it from other cancers with hormone receptors, such as breast cancers or prostate cancers, that if you cut the supply of the hormone to these cancers, the cancers will not grow very well. So in a way, that is a good control of these type of hormone dependent cancers. But then the major question is, we don't know who will benefit from this type of therapy and who would not. So, but we do want to find out the answer to this question, because obviously we don't want to expose patients to anti-hormone therapy and its associated toxicity if the individual with the disease is not going to benefit from the therapy. And of course, also, if there are patients that we know that would not benefit from this kind of therapy, then better alternatives should be sought for them. So that we want to find out the answers to this question. And then to this, we actually made use of a very specialized model system. So this is an example of how some of our model system can be used for research. So in this very specialized model system, we receive consented patient tissue, and then we use a specialized surgical technique to put the patient tissue, as shown as a white piece of tissue here, under the renal capsule of the experimental mice. And then under this condition, the patient tumor is going to be preserved very well. And then we're actually able to do a short-term experiment for 2 weeks. Basically, we expose the mice under either estrogen or without estrogen, and then to see how the tumor would actually react under these conditions. So then this is an example of a patient tumor. This is directly from patient tumor, actually exposed under the conditions that I've mentioned, either in the presence of estrogen or in the absence of estrogen. So I hope that you can see that in the presence of estrogen, the cancer cells are actually much more full and plump, and they look healthy. But in the absence of estrogen, they actually sort of shriveled up like raisins or grapes that's shrivel up, or dried raisins. So they don't look that healthy. And in order to help us to better visualize the difference, we perform a staining that's called Ki-67, as you can see with these brown dots here. Basically, what it indicates is that if you have more of these brown dots, it indicates that there are more of these growing cancer cells. And then you can see that in the absence of estrogen, in this case with this patient tumor, when there's no estrogen, then there is a drastic decrease of these brown dots, meaning that there's a drastic decrease of the growing cells. So there's a high chance that this patient tumor would actually respond quite well to anti-hormone therapy, i.e. that is represented with the absence of estrogen in this case. But as I also mentioned, not all the low-grade patient tissue would actually respond to this kind of therapy. And this is one of the examples. So as you can see here, that's in the presence of estrogen, the cells, again, they organize into this well-structured pockets of cells, and they seem to be growing quite well. But you also notice that in the absence of estrogen, they still look the same. And also, as I mentioned, to help us to better visualize the effect of the absence of estrogen, we also did Ki-67 staining. And as you can see, the amount of brown dots is actually very comparable between the case when you have estrogen or when you have the case that there's no estrogen. So by performing this short-term assay, we're actually able to pick out some of the patient tissues that would have a high chance to respond well to anti-hormone therapy. And there's also, we can also pick out the cases that would not respond well to anti-hormone therapy. So through studying the differences between these tumor samples and combining the clinical data that we can access to, we trust that we can actually formulate a set of parameters that will help us to predict which patient or which low-grade cancer patient will actually benefit from anti-hormone therapy and which patient will actually not benefit from the anti-hormone therapy. So this is our goal with this project. But I would also like to suggest that with the example that I've given you, there's a lot of implication in personalized cancer treatments. And as you may know, for rare cancers, it's very difficult to find the right treatment for these patients. And some of these patients perhaps today is enrolled in some of the personalized anti-cancer program in larger centers, in which case they would actually consent to give some of their the tumor samples. And the tumor samples would be analyzed. They will go through genetic testings to analyze perhaps the mutational landscape of these tumors. And then with that information, several drugs would be suggested that potentially they may have an effect on these tumors. And then these drugs will be suggested to a tumor board of clinicians and scientists. And then with the best knowledge, they will pick out the drug that would probably work the best for these very rare kind of cancers. But what is missing is that so far, there's no good biological assessment, but with the short-term assay that I have just described, this might actually partially provide the answer. And as you can imagine, for example, if a tumor that doesn't have known good treatments, with the genetic analysis, potentially would suggest 3 to 5 potential treatments. And you can imagine if we are able to subject the patient tumors directly to these 3 to 5 suggested treatments and then pick out the best. So we will be able to best equip to find the proper treatment for the patients that would not normally have a very good kind of treatment. So this is also an example of how we can actually use some of the models that we have been developing in the lab. And so with that, then I would like to hand it over back to Dr. Carey to conclude our presentation.
Well, thanks very much. You can see that I'm privileged to be able to work with such a fine group of individuals, and we're pleased to be doing this work on behalf of patients and individuals with low-grade serous cancer. In line with our title slide, I think we've been able to show that these models that we've developed from patients who've contributed their tissues and for this type of research that we're doing our best to leverage that to accelerate the research that we're doing. And I think in low-grade serous cancer now there's perhaps 3 new treatments on the horizon, which is really, really encouraging for us. And we just have to continue to do our work. We've been privileged to witness an incredible amount of advocacy on the part of public fundraising groups like Ovarian Cancer Canada and patient advocates like Jane Ludeman here, who started the Cure Our Ovarian Cancer program and sponsors the International Consortium on Low-Grade Serous Cancers, and then we've been privileged to have people like Donna and Karen who have helped us with our grant writing and helped us move some of our projects forward. And we really appreciate their input, and people like Amy, whose family has also contributed to fundraising initiatives in low-grade serous cancer. And then a relative of mine, Janet Cottrell from Toronto, who Janet has contributed to our work as well. And Janet's contributed to other ovarian cancer programs in Toronto in the Sunnybrook area. And Ellie Mayday, who some of you may know, who's a plus size model who really made and enhanced awareness in low-grade serous cancers through her public her public disclosure about her illness, and she really heightened awareness of the disease and has really helped us focus some of our fundraising efforts on improving care. So we've done a lot of work, but we still have a lot of work to do. And when it comes to the work for the patient, we really want to continue to focus our efforts on finding new treatments and creating better support systems for our patients. We recognize that there's huge financial challenges for patients with cancer, and there's really many facets of work that we can do to help better treat and manage this disease. So we're committed to doing that, and thanks for your attention today.
I want to thank you all for that. It was wonderful to see all of you together and to hear from most of you. We just have time for one question. So low-grade serous can be so confusing for patients because the standard of care varies globally. Many countries treat low-grade serous with chemotherapy. Under what conditions would you recommend chemotherapy? And is there a specific therapy that has shown efficacy?
And that's a great question. I mean, there's right now, there's a large randomized trial that's being conducted because we still have a lot of work to do to sort out what the optimal treatment approach is. And I think the answer to that's going to be that it'll be somewhat different depending on the type of low-grade serous cancer one has. So yeah, the study is comparing chemotherapy with what we call maintenance anti-hormone therapy. So the patients get chemotherapy first, and then that's followed by maintenance anti-hormone therapy like letrozole, versus patients just going on letrozole. And so that study is accruing relatively well, and I think it's going to give us more answers with respect to how best to choose patients for the right treatment. And I think that's where we're going, because what we're seeing is that, like, hopefully with these other treatments that are going to come online soon, that there'll be specific patient populations that will benefit from treatment A. And there's some new drugs called CDK4/6 inhibitors, or palbociclib is one example of that drug. And they may be beneficial in a certain subgroup of patients versus standard chemotherapy in others, versus hormonal therapy or combinations. So I guess the answer to the question is I can't answer the question right now. We're in the midst of trying to better understand which treatments will benefit which group of patients, and then how to best sequence those treatments for patients.
Okay, great. Thank you. It's exciting though that there is a lot on the horizon. So thank you. And I want to thank you all again for joining us tonight from Vancouver. It really was great to see so many people involved in the presentation. So thank you very much. As Tanya mentioned earlier in the evening, we take a multi-pronged approach to research to ensure that all types of ovarian cancer, even the rare forms, are prioritized to ensure that everyone benefits from the best available science as quickly as possible.